Hi and welcome to this biology revision session on cell division. So chromosomes are found in the nucleus of a cell and they are made of DNA. Each chromosome carries on it a large number of genes, and each gene is a section of DNA that codes for a single protein. In our body cells, chromosomes are normally found in pairs, and as humans we have 23 pairs of chromosomes in each of our body cells. That means each of our cells contains 46 individual chromosomes. The cell cycle is the process by which our cells replicate themselves. During the cell cycle, the genetic material is doubled and then divided into two identical cells. Before we can start with dividing the DNA, though, we need to increase the size of the cell and the number of subcellular structures within it, so the number of ribosomes and mitochondria, for example. The DNA then replicates to form two copies of each chromosome. So instead of the 46 normal chromosomes, you would have 92 chromosomes at that point in the cell cycle. And then in this process called mitosis, one set of chromosomes is pulled to each end of the cell, and the nucleus then divides. Once the nucleus is divided, the cell membranes and the cytoplasm divide around them to create two identical cells. And those will then have to grow and carry on the process, which is why we call it the cell cycle. So why is mitosis important? Well, it produces cells that are genetically identical to the original cell, which is useful because it enables us to grow and repair without showing differences in our genetic structure. Stem cells, we talked about briefly in the last session, are undifferentiated cells, so cells that don't have a particular function, and they can turn into multiple different types of cell through differentiation. Embryos are made up of stem cells because they will need to be able to form all the different types of cells in the human body. But adults also have some stem cells. For example, the bone marrow contains stem cells which can produce different types of blood cells. In plants, meristem tissue can differentiate into any other type of plant cell, and that happens throughout the life of the plant. Because stem cells can turn into other cell types, they could be used to help with treatments for conditions such as diabetes, where the pancreatic cells aren't working properly, or paralysis, replacing nerve cells, for example. In the process of therapeutic cloning, an embryo is produced which is genetically identical to the patient. The stem cells can then be used to produce tissues or organs which are genetically identical to that patient. And because they're identical to the patient, they won't be rejected by the patient's immune system. So if you needed a replacement kidney, for example, if you have a kidney transplant for someone else, there's always a risk that your body will reject it. If you were to create one using therapeutic cloning, then there would be no chance of rejection. However, there is a potential risk of viral infection using this technique. Another problem is that some people object to therapeutic cloning on ethical or religious grounds, saying that once you create an embryo, it should be allowed to grow into a full human being, or saying it's playing God. Less controversial is the use of plant cloning, and clones of plants can be produced quickly and cheaply using those meristem cells. This can be used to protect rare species by replicating them rapidly, but it can also be used to rapidly replicate plants with desirable characteristics. For example, if you have a crop with disease resistance, you would want multiple copies of that plant so that you can have your whole yield of crop having that disease resistance. Okay, that's all for today. I'll see you next time.